Hi everybody, this is Professor Kleiman and Alex Rack. Today we will talk about the experiment for DNA transformation and selection of colonies with the transformant DNA. Alex?
Okay, so this is a really good experiment because it kind of ties together the previous three DNA-based experiments that we've done, which are DNA digest, DNA ligation, and PCR, polymerase chain reaction. So today we're going to be working with a plasmid called PUC8, and this plasmid has a couple of genes of interest. The first is ampicillin resistance. So any bacteria that has this plasmid will be able to grow in the presence of the antibiotic ampicillin, which normally bacteria cannot. Second, we have LAC-Z prime. Now, this is one half of the gene that encodes for the protein beta-galactosidase. Beta-galactosidase is an enzyme that will degrade and cut apart different types of sugars. What's important is that PUC8 plasmid only has, as I said, half of this gene. This is the amino half, so this is the front half of it. Normally, the E. coli we'll be using only produce the second half of the protein. Now, if you only have the first half, or if you only have the second half, it's non-functional. But if you have cells that are producing one half, and then you introduce this plasmid, which allows them to produce the second half, even though they're two separate parts, they can still come together and be fully functional.
That's what we call alpha complementation.
So when we have both halves together, you have a fully functional enzyme that can degrade those sugars. Also here, you'll see MCR. This stands for multiple cloning region. This is a region that's been inserted in this LAC-Z gene where multiple restriction enzymes can cut. And in this experiment, we're going to be using DNA that's been cut in the multiple cloning region by EcoR1. So moving on to just a breakdown of the experiment we'll be doing. We are starting with a plasmid that's already been cut. It's been linearized, and it's been linearized by using a restriction enzyme, just like we did in the previous experiment. We're going to be adding a short piece of DNA and seeing if we can insert it into the plasmid. Now, where would this piece of DNA come from? Obviously, you can purchase it, but the easiest way to get a piece of DNA, if you're trying to get a specific gene of interest, would be through PCR. Just like the experiment we did before using polymerase chain reaction, you can amplify a specific gene of interest, and once you have that, you need to be able to make more copies of it, or even insert it into living organisms for experiments. So this would be a perfect example of how to do that. So we're going to have two setups, an experiment and a control. In both of them, we're going to be taking the plasmid that's been cut in the MCR by EcoR1. In only the control, we just have the plasmid that's been linearized, and in the experiment, we have the plasmid and that piece of DNA. We're going to combine them together with ligase, the enzyme that attaches cut pieces of DNA, with ATP. This is very important because ligase is an enzyme that requires ATP for energy in order to function. It's going to mix these together in separate tubes, and then we're going to incubate it at room temperature. This can go on for 30 to 60 minutes. At the end of that incubation period, we're going to have a couple of different results. In the control, where we just had plasmid by itself, any successful ligation will simply seal up that plasmid. So we'll be starting with exactly what we have here. However, in the experiment, we're going to have two different types of outcomes that are possible. The first is going to be the same, just as in the control, where our plasmid will just self-anneal. It will ligate into a full plasmid. But the second outcome, which is what we're looking for, is our piece of DNA will be inserted into the cut site when it gets ligated. Now,
an important thing I want to clarify, the insert, the little piece of DNA that could be the PCR product, has to be digested with the same enzymes. So in this case, will be also digested with EcoR1.
So now we're going to take these plasmids and introduce them to E. coli. This is our transformation step. We're going to do what's known as a heat shock treatment, where we mix the DNA in some cells, and we bring them to a high temperature for a brief period of time, then a low temperature for a brief period of time, and then we allow them to kind of heal at room temperature for about an hour, and then we plate them. This simply means we introduce them to an agar plate where we allow them to grow. This heat shock treatment weakens the cell, and it allows the plasmids to be taken up into the cell. Now, one important part about this insert is because it gets inserted into the LAC-Z gene, remember, this will now disrupt the function. It'll disrupt the ability of this half of the enzyme to work, which means even if the cell has now taken up the plasmid, it's no longer going to produce a functional enzyme. So in the plates that we're growing the cells on, we have a special sort of sugar called X-gal, where once it's digested by this enzyme, it forms a blue color. Normally the cells form white colonies, but any successful degradation of X-gal will lead to a blue product, which makes the colonies blue.
Remember, this is like a substrate analog to lactose. You remember beta-galactosidase degrade lactose, so this is a substrate analog that, as Alex mentioned, will give you blue.
So what we expect to see after growing these cells overnight, our control colonies should all be blue. And our experiment colonies should be a mixture of blue and white, depending on whether or not the plasmid has taken up the insert. Now, another good thing about this setup is that we need a way to ensure that any plasmid has been taken up, because we don't know, if there's an insert or not. But we know if there's a plasmid because of the ampicillin resistance gene. Again, the E. coli will die in the presence of ampicillin, but whether or not the insert has been taken up in the plasmid, this plasmid has an ampicillin resistance gene in it, so the only cells that will grow in the end have taken up the plasmid.